barcoding them. Our model probably does not recognize them, but we are on route.
Thank you, Peter. So, okay, Tuka, he's coming. You okay? You're looking around anxiously. Yeah, over to you.
Hello, everybody. My name is Jarrett Blair, and today I'll be talking about combining computer vision and DNA barcoding for high-throughput invertebrate specimen processing. So before I get into the meat of the talk, I just want to give a bit of framing. So for the past 10 or 11 years, Toga's group has been doing pitfall trapping in the south of Greenland around a small village called Narsassuaq. And there we have 140 traps sampling roughly 3 to 5 weeks each summer. And doing some rough math, rough estimate, I would say that we have around 500,000 insect specimens stored in ethanol now in Denmark that need to be processed. And doing this completely manually would seem like a almost insurmountable task. So this would be a project that would seem like a good candidate for automation. And fortunately for us, we have the BioDiscover machine, which for those who don't know, is a machine designed to quickly image bulk arthropod samples. And it does so by imaging specimens as they fall through an ethanol-filled cuvette. On this machine here, you can see there's a little opening. The specimen is dropped through that opening. It falls to the ethanol-filled cuvette. It's imaged by 2 cameras. I can show them here. And these cameras will image the specimen as it falls to the ethanol, so you're getting those 2 angles as well as any rotation. And then it's dropped into a vial underneath the machine. And recently we've developed this carousel mechanism that goes underneath the machine that can allow you to automatically switch out the vials. So we can sort the specimens by their species identity, their size, etc. And so this is a really great machine for our purposes. But if we wanted to say sort these species to species level through an automated methods like computer vision, we'd likely face some challenges because computer vision is not entirely perfect. For one, there's a long tail problem. So that's where most species exist in this long tail of within a community where there's very few individuals, which makes it difficult to train a computer vision model to classify them. There's also cryptic species like, say, this fungus gnat that are virtually indistinguishable based on their external morphology from other related species. And so you can't expect a computer vision model to necessarily distinguish these species by itself. When faced with these challenges, we'd often think of turning to a human-in-the-loop method to then sort the rest of these. But humans can also face similar challenges. For one, identifying species in that long tail requires a diverse taxonomic expertise. That those species are going to come from a wide variety of taxa, and so it's unlikely to find a single or even a few taxonomists that could identify them all. And cryptic species often take longer to identify because you're having to, say, dissect their internal genitalia, and again require highly skilled taxonomists. So this isn't necessarily a job for, say, a handful of bachelor students. You're going to need, some highly trained people, which may be difficult to get access to. So if we're not using a human-in-the-loop method, what could we use? And as you may have guessed by the title of the talk, I think DNA barcoding complements computer vision really well, especially with some of these challenges. First, DNA barcoding is not dependent on morphology, so those cryptic species can, at least a good number of the times, be distinguished using these molecular approaches. There's also huge public reference databases, so species in that long tail are more likely to be captured in, say, a database like BOLD, which has over 300,000 insect specimens cataloged. If those specimens, or if those species aren't cataloged on BOLD, you can also create your own custom reference databases with a relatively few number of voucher specimens, or use operational taxonomic units or OTUs to group putative species based on similar barcodes. So for those in the audience that might not be entirely familiar with what DNA barcoding is, I'll just do a brief rundown here. So you would start from an individual specimen and extract its DNA, either using homogenizations, that would be, say, blending the specimen, or using cell lysis, which is a bit gentler and just releases DNA off of it. Once you have that extracted DNA, you would amplify it using PCR, so you make a whole bunch of copies, and then pass it through a sequencing device to get the actual genetic code, usually from a particular region of the DNA. You then blast that against a reference database like BOLD, so that's just comparing the reads that you got from your sequencer against that database. And then hopefully, if everything worked, you get a species ID. Now, in the past few years, there have been advances in sequencing technology, specifically with the third generation sequencers, that allow us to do all this in a very highly parallel way. So it's much more efficient. And this is what we're using in our lab. And I'll just go over what that kind of looks like here. This is also called DNA mega barcoding, so high throughput DNA barcoding. And it starts with the extraction process. So when we take a sample, we would sort our specimens into these 96 well plates. We leave one well empty, so we have 95 specimens at a time getting their DNA extracted. If the specimen doesn't fit into the well plate, we just take a leg off or some other part, but most of them do fit. We then submerge the specimens in an alkaline lysis buffer, which gently releases DNA off the specimen. That then goes through an extraction process where we then separate the DNA from the specimen, and the specimen can be then stored and used later. And then we take that extracted DNA and amplify it using PCR. At this stage is also molecularly tagged, so we can link that DNA back to the specimen which it came from, or back to the well which it came from, which then tells us which specimen it came from. And this is important for the next step, because this is where we get really parallel, where we take those plates that we extracted and amplified the DNA, and we pool them all together into one tube. And at this stage, it's okay for the DNA to get mixed around again because it has those molecular tags. So in our pipeline, what we've been doing is taking about 50 plates and pulling them all together. So we have around 5,000 specimens all going through this at the same time. You then amplify the DNA, or not amplify, but purify the DNA, and then drop just a 75 microliter drop of it onto a third generation sequencer, like a min-ion device, which will then give you your DNA reads that you can blast. And a note for this too, these sequencing devices have become a lot cheaper and a lot smaller over the years, so it's much more feasible to do this all in-house without having to ship off your samples to some sequencing lab. Our min-ion device costs around 3,000 euros, just for reference. But then you might be asking, if DNA mega barcoding is so efficient and it can get us species level IDs, why even bother then with the computer vision side of things? Why not just do everything with DNA? And to that, I would say that the complementarity of computer vision and DNA kind of goes both ways. So with our biodiscovery machine, we can go through 2,000 or more specimens a day, dropping them through our machine at effectively no consumable cost. There's a bit of that ethanol involved, but outside of that, it's just the cost of the human labor that's dropping the specimens. Whereas with our DNA barcoding setup, it's about 10 times slower, so 200 specimens a day at 25 cents consumable cost per specimen. These numbers also do vary a little bit depending on how how many specimens you're processing at a time, but these are sort of the rough estimates to give you an idea. And then, as we discussed earlier, this complementarity goes both ways, where computer vision has the aforementioned problems as well. So with this in mind, then the question sort of becomes, well, how do we combine computer vision and DNA barcoding in a way that leverages each of their strengths while sort of, limiting their weaknesses. And for that, we have sort of thought of two approaches. The first is DNA-assisted model training. So with this, we would take our raw bulk samples, pass them through DNA barcoding, which will then give us our group specimens at the end. Again, the barcoding process is non-destructive, so these specimens pretty much look like the way they did at the beginning. We can then pass these through Biodiscover for imaging, which gives us a labeled image data set at ideally the species level that we can use to train a classification model. Then once we have that classification model, we can sort of flip this and do, we could call, say, DNA in the loop model validation with, again, starting from your raw bulk samples, doing imaging, going through classification, and then just really using the computer vision to target your known, more easy to classify, probably more abundant species. Those will be sort of sorted out into their own known groups. And then things that you could say maybe you can only classify at family level or belong to some, cryptic species complex. Those go into an out group that you then pass through barcoding. And at the end, if everything worked perfectly, you could get, pretty much your entire sample identified down to species. So how far are we? We're still fairly early on in this process. So before we began, Orcus didn't really have a DNA barcoding lab. We had a few PCR machines, but we had to get the rest set up. So that's set up now. We've barcoded around 9,000 total specimens. So we feel like that's a decent enough pool to start doing our imaging, maybe train a first model, probably realize that model is not very good, and then do the process iteratively a few times. So I'd say we're at around step 4 on this particular diagram, and then again in the future, ideally we're processing most of our specimens with computer vision because it's more efficient and cheaper, but we have DNA barcoding as a backup. With that, I'd just like to thank everybody that helped me on this project, especially Sven Lemke, who was a big help with actually barcoding and getting all that stuff set up. So thank you.
I love the term DNA in the loop as well.
Yes.
Fantastic. Cool.
Yeah, I heard the word sorting a lot. So I was wondering how much human labor is actually involved in that, or if you automated that to some extent.
With in the DNA process, like yeah. So for the Biodiscover, it's fairly straightforward. It depends a little bit depending on how your sample is stored. Let's say in a general case where you just have a pitfall trap vial, you would just sort of dump that out and just start. A human picks it up and puts it in. We do have a robotic arm as well for the Biodiscover that we've been trying to automatically do that as well. It's slower though than a human. It has the advantage of that you can like leave it running. But we've still found that humans are faster that way. For the DNA barcoding side of things, it's a little bit more involved because you're having to put the individuals into those well plates. So it's just a bit more precise. Really, in that stage, it's the most time-consuming part of the process is actually putting them into the well plates. There's a lab